Our last speaker of our session 3, focused on data analysis, will be my colleague Gang Yao from GSK. Gang has been at GSK for over 15 years, starting in 2005 as part of Praxis. And before he was at Praxis, he also worked at Biogen and Arcule, and he obtained his Ph.D. From Boston University. Gang's a senior leader in the group, having a team focused on hit lead chemistry, off-DNA, hit confirmation, and he's going to tell you about a number of the projects that we've worked on at GSK, as well as examples from the literature at other organizations, focusing on strategies for optimizing DEL hits and things that you have to consider that might be different than following up on HTS hits. And I see that Gang is trying to get something and getting settled there. So I think he's settled now. So yeah, so I'll give it over to Gang to tell you about some of these case studies.
First, I'd like to thank Lisa for the introduction, and I'd also like to thank the committee for giving me this opportunity to present the work here. So the topic of my talk today is a strategy for hit-to-lead optimization of DEL-derived hits. First, a few words about ourselves. We're the ELT DNA-Encoded Library group located in Cambridge. We're mainly focused on generating hits for the internal GSK programs. In GSK, here's the outline of my talk. First, I'll give a brief introduction to the DEL workflow, and then I'll show you the unique features of DEL hits and step and the common techniques used in DEL hit follow-up. And the last, I'll show you several case studies which highlights how those techniques are used in the following up DEL hit. So in GSK, we use DNA-encoded library together with the other technology as a multi-prong approach to generate hits. Comparing to other commonly used hit ID platform, DEL has the advantage of being able to access billions of molecules with low cost. However, it does require the off-DNA synthesis for hit confirmation, which may not be the case for other hit ID platforms such as HTS or phenotypic screening. So there's a special need for making off-DNA compounds for this platform to work. Here's some DEL technology 101, which has been, some of this part has been nicely described in the earlier talks. So first, there's several key steps involved. First, the screening of the DEL molecule against the target protein. This is mostly done through affinity screening. And after the selection, the selected DEL molecule, the DNA tag was amplified and sequenced and translated into structures of the putative small molecule binders. And after that, the on-DNA hits was synthesized off DNA and evaluated in the relevant biochemical and the cell assay to generate qualified hits. So the talk of this part is mostly focused on the last part of the DEL platform. So here are some characteristics of DEL hits. So I'm mostly focused on the DNA recorded chemical synthesis. There are many flavors, and the DNA recorded synthesis method most commonly used in the pharmaceutical industry. So it's obvious that each DEL molecule contains a DEL tag that records the history of synthesis. And each tag, because of this, each tag may encode multiple products. And for example, including the full-length molecule, intermediates, and maybe byproducts. And there's a wealth of on-DNA SAR information available due to the large number of DEL molecules that can be screened. And also, this information can provide further information to decipher the real binders, and also provide clues on the hit-to-lead, subsequent hit-to-lead effort. And the last, just want to say that because the DEL tag, which is a big DNA molecule, very water soluble, based on the all the available crystal structures of the DEL hits, they always point towards solvent, which provides some information on how DEL molecule may bind, sometimes useful in the docking study. Also provide handle to optimize physicochemical properties, and this also make DEL hits ideal precursor to make other bifunctional molecules. Here are some common DEL hit conformation strategies, and direct off-DNA synthesis, which has been used since the beginning of the DEL technology, which provide high quality readout, and however, it has a low throughput. And more recently, there are several papers published on high throughput hit conformation. There's 2 papers, one from WuXi, the other one from Pfizer, which utilize on-DNA, high throughput on-DNA synthesis. And the hit was confirmed using, the on-DNA hit was confirmed using mass spec. And more recently, GSK published a paper which used cleavable linker, and after the high throughput synthesis of the on-DNA molecule, the warheads was cleaved and detected using the ASMS. However, both technologies still need final confirmation, still need to make the hits off DNA as singletons. Here are the common steps and the technologies used in hit follow-ups. So once the enriched on-DNA binder was identified, they were usually clustered and then further triaged based on the selectivity profile of physical chem properties. And the top on-DNA hits chemical series was then confirmed by making off-DNA compounds to generate off-DNA hit series. And this was then further followed up in the hit-to-lead process to generate leads for the MedChem program. During this process, it has been shown the lipophilic efficiency is a good metrics for monitoring this progress. So you can see there in the following talk, there's always a improvement in terms of lipophilic ligand efficiency during the process. There are 2 papers, a nice paper recently published that study the trend of hit-to-lead trajectory for ELT hits. And overall, it shows, the ELT hits and HTS hits and fragments, they may follow a different trajectory. For example, ELT hits often, because of CombiChem nature, you get a very potent hits, but maybe has a less desirable physical chem properties. So there's a significant amount of work, effort on optimizing the physical chem properties in addition to the potency. But overall, no matter where the hits came from, you always end up in the same good space with a good potency and good physical chem properties. So here are some trend from some recently published ELT hits efforts. You can see again the trend is quite obvious. You can see there's a improvement during the hit-to-lead process. There's an improvement in both potency as well as physical chem properties, mostly through a reduction of C log P and overall improvement in terms of ligand lipophilicity efficiency. Here's a reference, for this the papers in the previous slides, and some of this will be used in the following talk to give you case studies. to show you how these techniques are used. The first example is IDO. IDO is a rate-limiting metabolic enzyme that converts the essential amino acid tryptophan into downstream catabolites known as kynurenin. IDO has a potential to restore immune function in a number of indications, and thus has a therapeutic applications in cancer and infectious disease. In this case, Indo hits was on-DNA Indo hits was identified through selection, and you can see here the cycle 1, and which is linked to the DNA tag, is variable as not selected. So off DNA synthesis generate this compound, Indo compound, which truncate out the whole piece, cycle 1 piece, as well as the DNA linker. This compound shows good potency. However, it has a high in vivo clearance, presumably because the electron-rich Indo rings. SAR study indicate the T-butyl group can be replaced with ethyl carbamate with improved potency. Further assay study, assay work, basically replacing the Indo-ring with a more metabolic stable indazole-ring result, and adding another substitution on the phenol-ring result in this compound, which shows good potency and with improved PK profile. And however, because the C-log-D of this compound is still a little bit high, so which result in low solubility, and the prodrug approach was then used to make this compound. This compound shows a high bioavailability and high solubility, and has a low predict human dose of 60 mg. So my next example is Z-alpha-1 antitrypsin misfolding corrector. Anti-alpha-1 antitrypsin deficiency is characterized by the misfolding and the intracellular polymerization of mutant alpha-1 antitrypsin protein within the ER of hepatocyte. And individuals with a severe alpha-1 antitrypsin deficiency led to early onset of emphysema. So small molecule corrector of Z-alpha-1 antitrypsin folding block can have a application as an oral drug for the treatment of alpha-1 antitrypsin deficiency. Again, on-DNA hits was identified from the Indo library, 3.6 million compounds, and the BB1 was not selected. In this case, also when we made the off-DNA compounds, the BB1 as well as the DNA linker and the DNA tag was cleaved and generate this small molecule compounds, which shows very good potency and good ligand efficiency here. And after resolve the stereochemistry, it shows that 1s, 2s, 2r diastereomers is the most potent binder here. So further optimization of replacing the Indo-ring, which is as in the case in the previous example, is metabolic liability with benzoyl oxazolone ring, result in this compound here, and which shows a little bit improved potency, and which result in a little bit better ligand LLE. And crystal structure of this compound was resolved, which shows there's hydrogen bonding between this carbamate moiety with a tryptophan through a water molecule. Further optimization by modification of this part of the molecule, resulting in this lactam moiety here. Also by moving around this phenol ring, benzene ring, the potency improved a log. You see a log improvement in potency. Also, more importantly, and you can see there's a good cellular activity now observed. X3 structure was solved, which indicate by moving this moiety around, this lactam ring moiety pick up additional hydrogen bond interaction instead of with this indole you form a hydrogen bond with the glutamic acid and the lysine, which account for the 16-fold increase in potency. And this compound, turned out to be a highly potent cellular active and selective alpha-1 antitrypsin corrector. So my third example is discovery of clinical candidate RIP1 kinase inhibitor for the treatment of inflammation disease. And in this case, on-DNA compound with oxazorin was identified from 7.7 billion member DEL. And by truncation of the DNA linker part, we identified this compound on the right, which shows a very good potency. However, it has a low oral bioavailability, presumably because the metabolic liability of this oxazorin. So by doing a scaffold hopping, we identified this compounds, triazo compounds, which reduce the ClOc D significantly. Also, it improved the oral exposure significantly. So by simply just replacing the oxazorin with the triazorin, we identify a clinical candidate, RIP1 clinical candidate shown here. So in the next, I'll just show you some examples of two examples of BET inhibitor identified through DEL. The BET family contains 4 related proteins, namely BRD2, BRD3, BRD4, and BRDT, that act as epigenetic readers, which has a broad application in the transcription activation. And each BET family member contains 2 tandem domains. A difunctional BET family member is involved in many physiological and pathological process and has become an important therapeutic target for disease, including cancer and inflammation disease. In addition to inhibiting the both domains, and more recently, targeting one of the 2 bromodomains has begun to emerge in the literature. And these inhibitors may help to determine the role and the function of each domain, and may be able to help access if we can improve the therapeutic index for the BET inhibitors. So the first example is on the pan-BET inhibitor. The second one is on the BD2 inhibitor, specific BD2 inhibitor. In this case, first, after selection, we identified generic penzoimidazole hits from 170 million compound benzoimidazole library. And off DNA by synthesis by truncating the DNA tag result in this structure here, and several analogs was made and confirmed as BRD4 binders. And this series was driven by this dimethyl phenol moiety. But this phenol moiety is a potential metabolic liability because it's readily available, readily metabolized, for example, in the rat, has a very poor mouse in vivo PK data. So Then we examined we obtained the X-ray structure of this benzoimidazole hits, and by comparing the X-ray structure with our previous identified fragments hits, we realized we can replace the dimethylphenol is actually functional as a KAC mimetic. So we may be able to replace with a pyridine functional group here, which serve the same purpose, how form hydrogen bond with the conserved asparagine 140. So this indeed turned out to be the case. For example, once we replace the this phenol with this dimethylpyridone compounds, we find this compound shows similar comparable activity, but with a significantly improved physical properties. The C-log-D goes down from 3.2 to 0.94, which gave us more freedom to optimize other part of the molecule. Further optimization of the azopipridine moiety result in this compound, which has a dimethyl isopropyl group here. This compound showed improved potency and also showed a very good selectivity against similar bromodomains shown here. For example, BAS2A. The potency of this series can also be improved by adding a morpholin group in where the DNA vector was, so resulting a compound with improved potency, but in this case, it lost selectivity against the BAS2A. So by combining these two series, we identified this preclinical compound IBAT469, which shows a very good potency as well as PK profile and the cell activity, and which result in a pretty low human dose of 30 mg QD. So my last example will be on the highly selective, the identification of highly selective BAT-BD2 inhibitor from DNA library, DEL library. So in this case, we did a selection with a panel of BRD4s. First, truncate the tandem domain construct, and also the two mutant, one with a BD1 mutant, the other one with a BD2 mutant. After the selection and looking the selection profile, we identified one series shown here from 1.3 million DEL molecule library as potential BD2 selective hits. And after removing the DNA tag, this compound was made off DNA and confirmed as to be indeed BD2 selective. It has like 6.4 PSA50 against BD2 and 5.1 against BD1, overall 30-fold selectivity for BRD4. The X-ray structure of this compound was quickly obtained, and it was quickly realized that the dimethyl methoxy benzyl group also serving as a KAC mimetic. So in this case, using the same approach, we replaced this moiety with dimethyl pyridone compounds moiety fragments. We result in this compound, which with much improved potency, a big jump of 2 log improvement in terms of potency, and 4 log which result in a 4.2 increase in lipophilic ligand efficiency. Further improvement of this, the selectivity of this compound was achieved by examining comparing the structure of the BD2 and the BD1 domain. It was realized that the BD2 domain on the Z panel has more freedom to accommodate a bigger substitution. So we rationalized that by putting a substitution on the alpha group, we may be able to further improve the potency of the selectivity of this series. And this turned out to be the case. For example, when we add a methyl group on this on the alpha position, we improve the selectivity from 30-fold to 100-fold. But again, this compound still has a bit higher C log D, and maybe which may negatively impact its solubility. So further improvement both the selectivity and lipophilic log D achieved by, for example, introducing a bigger tetrahydropyran group on the alpha position, and introduce a nitrogen in one of the ring system. This result in compound GSK040, which has improvement of selectivity from 100 to 5,000. And this compound became a very good two compound to study the biology of the two bromo domains. So I'll skip due to the time, I'll skip the next two examples, but I just go show you, here's a comparison of the DEL hits versus lead. Overall, you can see you generally see improvement in terms of lipophilic ligand efficiency as well as potency. And this is again more examples, and here's some charts that shows this trend. And you can see in the during the hit to lead process, you would like to further improve the potency and also improve the physicochemical properties. And in terms of molecular weight, the trend is less clear. Sometimes you see a lower molecular weight, which is probably due to the truncation. That was a strategy applied for the hit to lead process. And sometimes you see increase in molecular weight, which are more used in the traditional MedChem process for improved potency and selectivity. And again, this is a very clear trend to show the during the hit to lead process, you see LLE improvement, and also sometimes accompanied by improvement in terms of ligand efficiency. In summary, here's some common hit to lead optimization strategies that's commonly used based on our experience and the published literature. First, you really want to take advantage of the on-DNA SAR information. As I mentioned, the information is very it's a unique feature for DEL hits, and due to the ability to screen billions compounds. And by utilizing this information, for example, usually when we do the analysis, we identify the parts that we call the selected part, and there's other part we call the non-selective part. And the selective part usually indicate there's a specific interaction with the protein. So it might be, so this is some part you really want to focus on, and to improve actually maintain this interaction and optimize the interaction for better potency, and especially trying to optimize some polar interactions. The non-selective part of the DEL molecule usually don't have any specific interaction with the protein. Sometimes they can be truncated completely. Sometimes they can be reduced, especially some lipophilic group that can be reduced to improve the physical chem properties. And again, the ligand LLE is a good metrics for this for the hit to lead optimizing process. And this improvement usually achieved by increasing potency while at the same time decreasing lipophilicity. And this, there are several common techniques used very often in the DEL-Derived process. For example, the scaffold hopping, which you use, I think is maybe more often than traditional MedChem. And also, there's several cases where polar interaction was optimized. For example, in the antitryptophan case, based on the structure, there was additional hydrogen bond interaction was introduced, which result in significant improvement in the lipophilic ligand efficiency. And the last, I just want to show, the structure-based design and the property-based optimization principle can play a big role in the DEL-Derived optimization process. So with that, I'll conclude my talk and take any questions. Let me see.
Okay. All right. Yeah, so we have a bunch in the chat. Hopefully I don't lose track of them. Okay, so the first one that came in was, in your case studies, was the DEL hit truncation done systematically, made every possible truncation and then data generated on each one, or did you have an idea beforehand of which building block could be deleted? How often do you truncate the initial hit in this way?
We generally have a indication which part of the molecule can be truncated by looking on DNA SAR. And as mentioned, there's a certain part of the molecule that we call selected, that's specifically enriched in the selection. Those parts we generally don't want to truncate or do modification before we confirm that. But there are other parts that's not selected. When we do the off-DNA synthesis, we often try to make different truncations, and sometimes we include the full length, and also we make the fully truncated, and sometimes we make like a partial truncated molecule in the heat conformation process.
Okay, and, it says, how many learnings to optimize the BRD4 hit were derived from the DEL output versus traditional MedChem optimization campaign?
There's certain information available, for example, especially SAR information that was available from DEL output. For example, the piperidine part was not selected, which give us information that it can be optimized, which used in the MedChem program. And the dimethylphenol part is selected, so that's why you want to put specific dimethylpyridine group there, not just anything, So those kind of information are used in the together with the MedChem information for the hit to lead effort.
Okay, I guess sort of along this line. So Fred Burst had asked the question, but then in a follow-up, because I guess you sort of answered it in your conclusions, he asked, do you find deriving pharmacophore models from DEL data useful in general?
Yeah, I think it's very useful. The only challenge is doing pharmacophore is much more complicated, maybe than the similarity, kind of model similarity clustering. So, we actually has published some paper we are trying to use like GSK, we call MGAP analysis. So we're trying to, identify some pharmacophores, so model, yeah.
Okay, and then let's see. Curious, how many variants or deletions of DNA attachment point do you typically explore?
We generally don't do too much exploration on the DNA attachment point because we think they are mostly exposed to the solvent, so it's not the most efficient part for interaction with the protein. So we usually, the first round, we try to keep some polar group, for example, the amide group on the molecule. And sometimes they have specific hydrogen bond interaction with the protein. Sometimes they don't. So in those cases, we can completely truncate those part.
Okay, and how are we doing on time? I guess this is a says general question for all presenters, but maybe you can give your thoughts on this. It says, what is the range of design, make, test cycles a DEL hit goes through before advancing to the next step? How does it compare to other hit finding approaches like HTS and fragment-based screening?
Yeah,
I guess it depends what the next step is. Yeah, okay.
yeah. For us, we usually don't do maybe 2 cycles. Usually one to 2 cycles before we transfer to the MedChem group for further optimization.
Yeah, I guess probably the answer depends on what the handoff is, and if you can hand off. And,
yeah,
but
right.
I would say that's accurate for what we do. Okay, and the last one, are there any trends in the ease of the hit to lead process when started from DEL hits versus HTS hits? I guess it depends.
Say again, what's the ease?
Are there any trends in the ease of the hit to lead process when starting from DEL hits versus HTS hits? I guess you showed some trends in the properties, but yeah.
Right. Usually, sometimes they are similar, but usually because the collection is different. For example, the HTS hits, at least for GSK, there's a cutoff in terms of properties, so they tend to be smaller and sometimes they are not as potent. So the ELT collection, because it's CombiChem, so it's more contained, more complex molecule. So we often find a molecule that's quite potent, but again, because they have some parts that's not very productive for binding. So those parts we try to truncate off when we make off DNA. So we spend more time probably than the in the physical property improvement than the HTS hits. Yeah.
Okay, and the Q&A box is clear unless anyone has any final questions. And I know we have a panel coming up with the latest advances in DEL's screening and other things. So I just want to thank everyone from the third session on data analysis for the great talks, Paul, Gong, Justin. Really enjoyed them. So thanks everybody. I'll sign off now too.
All right, thank you very much, Lisa, for a wonderful session, and all the presenters for session 3. That was fantastic.